up to introduce my soon-to-be ex-grad student, Ms. Vanessa Chappell. I wish she could stay longer, but she has to move on. But she has done more Westerns than John Wayne. Probably better at it too. She's done an incredible amount of work in the past couple of years. So we got a very compressed story here. Ms. Vanessa, rock and roll.
Hello, I'm looking at histone binding in mitochondria across eukaryotes. I worked specifically with cauliflower and also yeast for some of my other studies.
Down. I know it
Okay,
seems backwards.
there we go. Our hypothesis is once the mitochondria has been incubated with our histone samples, it induces cytochrome C release. And our hypothesis is this is a process that is conserved across all eukaryotes. We did cross-incubation studies, which also suggest that it is more suggestive that it is a conserved process because there was binding regardless of the source of histones. I also did a saturation study to show that if this is a receptor on the outside of the mitochondria, there are limited binding sites. So what prompted our study was a researcher named Histone back in 2013. He was working with a rat liver model, and he found that all 4 core histones bound to mitochondria and caused the release of cytochrome C. He was using HPLC purified histones. Our studies look at crude histone samples, so there were differences there that I'll discuss. And in mammalian cell, cytochrome C release is associated with apoptosis and other cell death mechanisms across all eukaryotes. Zanin, a fellow researcher, is also working with cauliflower, and she found H3 associated with the mitochondria in her samples as well. Her hypothesis is different than ours. She hypothesized that the histone molecules are actually being internalized inside the mitochondria, where we are hypothesizing that it's actually a receptor on the outside causing it. She also used different methods. Her methods were very crude. She used liquid nitrogen to freeze her samples, and then a mortar and pestle to grind her samples. We use much gentler methods, so our samples are less contaminated with histones. Preliminary data from a previous graduate student, Linda Major, she also found H3 bound to mitochondria. She did not have the time or budget to analyze any other antibodies, so we took on that task. But she did find that the binding to mitochondria caused cytochrome C release in cauliflower. She also looked at chlorophyll and found that it did not happen with chlorophyll. Levi Brewer worked with a yeast model, and he also found that cytochrome C was released after incubation with crude histone samples. He investigated all 4 core histones. Our H2A and our H2B and H4 were insufficient to show any type of result for that species. But he did find the binding with H3 causing release, and H2A bound but did not cause the release. Our methods, for our mitochondrial isolation, we use Percol gradient after differential centrifugation to remove any contaminants from the samples. Our histones, we simply protoplasted and then used a kit that was purchased from Epigentech to isolate our histones. Our incubation experiments, we're working with very small samples, so about 25 microliters of mitochondria were incubated with approximately 200 microliters of histones. We incubated them for 10 minutes and at room temperature, and then centrifuged them down and analyzed both the pellet, the mitochondrial pellet, and the succinate. The results were then analyzed using the SDS page and immunoblotting. All right, so the guts. Our incubation studies, there's quite a few of them, so I'll try to go over these lanes. Each column, the lanes are slightly different, but standard ladder showing what the size of the molecule that we're looking for is over here to the left, and our controls are right next to that, and then our mitochondria, histones, and then our actual experimental pellet and supernate. So here you can see with our plant, we used cauliflower. So with H3, right here with our pellet, the pellet is showing that H3 is bound to our mitochondria at this point. Here you can see there's no H3 in the mitochondrial lane, so that's our fresh sample of mitochondria before incubation. And afterwards, there is a strong signal that it did bind to our mitochondria. And there is some leftover, that saturation study I was telling you about, our theory that it has limited binding sites. If there's anything left over, you will see that in the supernate. So here with H2B, there is a small signal. You can see it. You can see our histone sample here, and then here's our pellet. There's nothing left over in the supernate. And for cytochrome C, to show that the actual incubation did cause the release of cytochrome C. This plot here, you can see our mitochondrial pellet here, where the mitochondria has the cytochrome C still inside. Our pellet, it does have quite a bit of mitochondria or cytochrome C in, but here you can see the band where there was release. Here's our BSA control. We just use that as a control to show that some other substance will also cause this reaction to happen. So here, our mitochondria was incubated with BSA, and all of the cytochrome C is still within the pellet. Right next to there is our yeast studies. These studies were done by Brewer, and you can also see that with H3, it bound to mitochondria and did cause the release of cytochrome C here quite a bit. H2A bound a little bit, most of it was still left over in the supernate. Slight binding, it did not cause cytochrome C release. This is after incubation with H3. So next is our cross-incubation studies. So we looked at studies incubating different species with each other to see if it caused the same reaction. The idea was if this is a conserved mechanism, it will not matter the source of where the histones come from. It will still interact in the same way. So when we incubated cauliflower histones, or mitochondria with yeast histones, it did bind. However, there was no cytochrome C release. With cauliflower mitochondria and mammalian histones, it did bind, and there was cytochrome C release. I kind of expected the opposite to happen. Completely unrelated species there, so that was an interesting point. Then we incubated yeast mitochondria with plant histones. They also bound, but they did not cause cytochrome C release. With yeast in mammal, again, it bound, but no cytochrome C release. Next, we wanted to see how strong this bind was. Could you wash it off with higher and higher molar salt incubation? So our normal incubation buffer is approximately 0.2 molar salt. So we looked at all the way up to 0.8 molar salt. We looked at 0.4, 0.6, and 0.8. So here with the plant, our cauliflower mitochondria, H3 stayed bound no matter what we put it in. There's no residue left over in the supernate. These are alternating pellet, supernate, pellet, supernate. So you can see it is a very strong bind with H3. H2B, a little less. You can see small bands. It's not a bright signal. However, it starts at 0.4 molar to between 0.4 and 0.6. We were able to wash that off with a higher molar salt. With yeast incubation, it stayed bound, H3 stayed bound through about 0.6 molar salt. With our cross incubations, it even stayed bound at that point. H3 showing the highest signals staying bound. Here with our H3 cross incubation with cauliflower and yeast, you can actually see the residue in our supernatant. So you can see it being washed off. With our plant and mammalian, same thing as well. It stayed bound through about 0.6 molar salt. And H2A and H2B, you can also see a strong signal there, holding until about 0.6 molar salt. And then you can see it in the supernate starting to be washed away. Our cross incubations with yeast and plant, same thing. For H3, it stayed bound through 0.8 molar salt. And for our yeast mammalian, also stayed bound through 0.8 molar salt. In conclusion, histones, no matter the source, seem to stay bound, indicating this is a conserved mechanism. So it kind of upheld our theory. We've been tossing around the theory of maybe there's competition between histones, because with our crude samples, we're seeing strong signals on our lots that H2A, H2B, and H4 are present. However, when we do our incubation studies, we're not seeing it in the pellet. It's not disappearing, it's there, but is it a competition between histones, especially with H3 being the culprit? Is it the first one to the race, that sort of thing? So we're playing around with that theory. Also, the histone mitochondrial interaction between other organisms holding true with high salt concentrations. It's a tight bind. For it to be bound at 0.4, 0.6, 0.8 molar salt, that's a tight bind. The data also suggests that it's highly conserved across eukaryotes since we are cross-incubating with other species. We're not just looking at one species in particular. And we think that it's a mechanism that's conserved to signal to the cell that there's been DNA damage done, or nuclear damage, that sort of thing. It could be part of the apoptotic pathway for a programmed cell death. Nobody really knows about that process in all detail, but so yeah, our data held true that it bound and cause a release of cytochrome C no matter the source of our histones. Questions?
Thank you. Are there questions for Vanessa?
How would you like to see this research applied to real-world application?
Real-world application. I think it could really be like an epigenetic insight to maybe some therapeutics that could go on. If you knew cytochrome C release was happening with your mitochondria, if you could maybe protect that mitochondria in some way to protect it from histone exposure, like mitochondrial diseases. Or there's not a lot of time to save a patient if you need some therapeutic aspect to block that while you're trying to come up with a plan. I think that would be great. Anybody else?
Can you go back to the last one of your incubation?
Yes. The first one?
Yes.
Yes. Okay.
Okay, so you didn't expect to see the H3 in mitochondria alone, is that right?
No. So I'm seeing a seasonal trend with this. In summertime, there is no mitochondria in cauliflower samples. The later in the season it gets, when it's probably been on a truck from Mexico for a week, it's already starting a dying process. So it's hard to get fresh mitochondria from cauliflower plant if you don't know its history.
Well, so like for lane 3 on the H3 lot,
Right here?
it looks like there's a signal there.
Right here,
Yes.
small signal. So that's showing you that there's been exposure. We killed it. And we do everything we can to purify that and remove that through the protocol radio. But there's still there's going to be exposure there.
Because I just wondered in some of the other lanes, like the very last lane on the same plot where you're just getting a really wide signal, is it possible you're just getting a bleed over? Because in every case you have a really strong signal and a really weak signal.
Like right here, this is, this is the supernate. So yes, it could be washing off, but that's a really tight pellet. These are tiny little pellets, and we're taking about 10 microliters of that.
Yeah, I don't mean washing
Okay.
off, but I mean when you load, you're getting such a strong signal in each lane right before where you're seeing a very light signal. Like
Okay.
when I load into the well, sometimes a little tiny bit goes into the well.
Yeah, I see what you're saying. Of course, like user error is always a
Yes,
factor.
but it's like up there, your light lanes are a consistent pattern next to your really strong signaling. So I just wondered in places where you didn't expect to see a band, could that be a factor?
I would say no, only for this reason. When I'm working with the pellets from these incubations, it's about 20 microliters to resuspend a pellet, very small amount. The supernate from this is about 150 microliters. And you have to mix that with your running buffer to even get it to load into your sample. So could there be a tiny bit of that pellet that's in the supernate? Maybe. I think it's probably more residual from the pellet than it is from spillover. That's my theory, just from watching it, you know, in the lab. But anybody else?
Barbara?
So what were you doing literature studies or literature review? Did you come across the cell signal pathway that forms release of cytochrome C?
Yes, there's a lot of them. Back in 2003, Tsugusugawa did a research study using tobacco leaves, I think, and he found the same thing. He went into a lot of different aspects of cell death pathways. He looked at the BCL2 families and that sort of thing. It was a really in-depth study. We didn't have access to do something quite that in-depth. But he was also finding the binding happening in tobacco leaves. And that's not the only one. That's just the one that popped up first. The others that I mentioned myself. Anybody else?
Very good. Let's thank our speakers.